All right, well, it is 2 o'clock right now. So welcome back. Hello. This is the virtual Harmful Algal Bloom Research Symposium. My name is Lois Wolfson. I work at Michigan State University as an aquatic ecologist and outreach specialist, and I'm going to be the moderator for this session. This symposium is hosted by the Algal Bloom Action Team, which I'm a member of. And this team is a collaboration of water professionals, researchers, and educators from 12 states in the north central region of the US. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and University Extension within each state in the north central region. And you can read the various partners. Excuse me. This afternoon is the fourth and the last session of the symposium, and we will continue to focus on animal health and harmful algal blooms. This session will last 2 hours and will feature 3 presentations on emerging harmful algal bloom research from leading researchers across the north central region. We will also have a 10-minute break at 1:45 Central Time. Following today's presentations, we will host a 1-hour facilitated discussion on the topic of this session to discuss how we can better disseminate information on HABs. And this discussion will help facilitate the work of the Algal Bloom Action Team. Our team is working to develop a website of resources, including fact sheets and FAQs on HABs topics. Additionally, we plan to organize webinars, recording video content, and to continue to host this symposium in the future. However, to do that, we're seeking your insight in how we can improve the dissemination of materials and content areas of research for the public. Now, due to the number of people at the symposium and the platform constraints, an additional sign-up sheet was required for the facilitated discussion, and all the spots have been filled. So we do apologize for any inconvenience if you weren't able to sign up, but we've been truly amazed by the interest in today's event, and we'll make sure we can accommodate additional people in the future. If you do have any questions about the facilitated discussions, or if you're having any technical issues, please note this in the chat box, and our team will do their best to assist you. During this afternoon's talks, if you do have questions for the presenters, then please place them in the Q&A panel at the bottom of the Zoom screen, and I'll ask them to the presenters after their presentations as time permits. So before we get started, I'd like to take a moment and thank our sponsors for making the symposium possible. In addition to the support of the North Central Region Water Network and the National Network of Water Research Institutes, we'd also like to thank our champion sponsors, FlowCam by Yokogawa Fluid Image Technologies and Limnotech for their support. Okay, so let's get started. This afternoon, we have 3 great speakers on the topic of animal health and HABs. Our first speaker is Ellen Preece. Dr. Ellen Preece received her master's and PhD from Washington State University. She investigates cyanotoxins in freshwaters, estuarine, and marine ecosystems, and uses this research to apply to determine cyanobacterial harmful algal bloom risks to human health. And one important component of her research is to ensure that results are available to local, state, and federal groups to sustain the ecological integrity of freshwater ecosystems while protecting human users, especially underserved communities. I am going to stop sharing my screen and let Ellen take the podium. And her presentation today is on cyanotoxin accumulation in aquatic animals and food webs. So Ellen, it's The floor is yours.
Okay, thank you so much for having me. I'm looking forward to speaking about this topic. I've been researching the accumulation of cyanotoxins in aquatic biota for about 10 years now. And so I'm going to start this talk by giving an overview of the state of the knowledge of the accumulation of cyanotoxins in aquatic animals. And then I'm going to present 3 examples of my research over the past 10 years, including a current study. And I'm going to highlight some of the things that we have learned along the way. So one of the things that you'll see throughout my talk is me addressing the different analytical techniques that are used to measure cyanotoxins in aquatic animals and some strides that we have made in that field. And then all of my projects have also looked at how the accumulation of toxins in aquatic animals may lead to risks to human health. And so I'm going to address that in each of my examples as well. So to begin with, cyanotoxins have been documented in a number of different aquatic biota. They've been found in what I call finfish, or fish that are swimming like bass or salmon. And they have been found in benthic feeding fish, those that feed on plankton, and then piscivorous fish. They've also been found in a number of different types of shellfish, including mussels, clams, and crabs. And then various other aquatic biota, zooplankton, amphipods, annelid worms, amphibians, and reptiles are just some examples of that. So I looked at several reviews when I put together my opening slides here that are in the literature that have synthesized all of the information that we know about this over the last couple of years. One of these reviews was a 2018 review by Flores and others, and this focused on finfish. And they found that 90% of the studies from around the world were focused on accumulation of microcystins in the finfish. The study did note 2 studies that had documented bioaccumulation of anatoxin A, and then a handful on BMAA, saxitoxin, and cylindrospermopsin. And I'll state here, there have been a lot of saxitoxin studies in aquatic biota and marine environments, but very few in freshwater environments. Little research has been conducted on A and saxitoxin in freshwater fishes in lower trophic levels, according to a recent review by Christensen and Kahn. And then another review that focused on cylindrospermopsin by Scarlett and others found that the toxin accumulates in mollusks, crustaceans, and fish, but information is very limited on this toxin. So based on this background and my own experience, my talk from here on out is going to focus on the accumulation of microcystin in aquatic biota. I thought this was an interesting graphic from the Flores review. This is the finfish study, and they documented both peer-reviewed and gray literature microcystin accumulation in finfish. And as you can see, it's widespread. This is happening all over the world, which is not surprising since algal blooms and associated cyanotoxins have also been documented across the world. Microcystins, like I said, can accumulate in a variety of different aquatic biota. In this figure on the right by Pham and Utsumi, they compiled a number of different studies across the literature to try to see which groups of aquatic biota accumulated the highest amount of microcystin. They found that in their study that zooplankton accumulated the greatest amount, followed by finfish and bivalves. And I'm assuming that the fish study was on muscle tissue. Microcystins do target the liver, and that's where we generally see the highest amount of microcystin concentration in fish. But from what I could read from this article, I think this box here is about muscle tissues, so the fillets that people are eating. So what are some potential impacts to aquatic biota from microcystin exposure? It can impact the embryos hatching. It can result in reduced survival and growth rates of fish, cause behavior changes, and cause liver necrosis, as liver is the target organ for microcystin. And it has been documented to lead to fish kills. But it should also be noted that biota can purge the toxins. And so you may have a fish or another organism that has toxin in it, and it appears to be perfectly healthy. And some of these animals can depurate the toxin. One study by Gibble and others found that mussels had microcystins for at least 8 weeks after exposure, and then they weren't probably undetectable after that. So they do depurate them, especially the shellfish, but they can persist for a while after exposure. So as I mentioned, I think that the analytical methods for measuring microcystin in biota is very important. It's really hard to compare microcystin concentrations across different studies due to the variety of analytical methods and extraction methods for pulling the toxin out of the specific biota. So the two most common methods that are analytical methods used to measure microcystins in aquatic biota are ELISA, which is an enzyme-linked immunosorbent assay that colorimetrically detects the toxin, and then liquid chromatography coupled with mass spectrometry. There are several other methods out there, but I'm not going to get into detail on those since these are the two most common methods for measuring the toxins. Flores confirmed that in their study that most of the literature that they reviewed people were using some form of ELISA to measure microcystin and other cyanotoxins in aquatic animals. Now, one of the issues with ELISAs is that they can be susceptible to matrix effects. There are a number of different ELISAs that are both commercially available, and then people can also make their own ELISAs in labs. And so depending on which type of ELISA you use, it can impact the toxin detection. So for example, there's over 200 microcystin variants out there. Each of them, unique to all microcystin variants, is this ADDA side chain that I think we have heard other speakers discuss. And there are certain ELISAs that are designed to specifically measure that ADDA and therefore measure all potential microcystin variants in a sample. Conversely, there are other ELISA kits that are targeted just for microcystin LR, and although they can have cross-reactivity with other microcystin variants, they may be less likely to measure all of the microcystins in a particular sample. And so depending on which type of ELISA a person is employing, it can impact the amount of toxin that is measured in a particular sample. Then LCMS is very specific and sensitive. However, there's only a limited number of standards that are commercially available. As I mentioned, there's over 200 microcystin variants, and I think maybe there's about 20 standards available, 20 different variants. So you can potentially underestimate the toxins, but you're less likely to have this cross-reactivity or matrix effects that you can have with ELISA. And then a method, the MMPB method is a method that can measure all microcystins with LCMS. Now, let me back up and say that ELISA is targeting those freely available microcystins, and so is much of the LCMS work, but a lot of the toxin can covalently bind to different tissues, and the MMPB method is able to measure both the free and the covalently bound toxins and measure all different variants. So why are more people not using that? It's expensive, and there's really limited labs out there that have the expertise and the equipment to do such an analysis. All right, now I'm going to get into my first research example. This is microcystin in Washington State Lake finfish. That is a study from 2008 to 2011, so one of the first studies that I worked on that addressed this issue. We collected fish from 6 Western Washington lakes with known cyanobacteria harmful algal blooms and collected a variety of different fish species. Just for people that aren't familiar with this region. This is the Puget Sound, and where my cursor here, this is the Seattle area. And so you can see where the lakes are relative to Seattle that we were sampling. We caught fish by hook and line, electroshocking, and gill nets. We looked at muscle, skin, gut, and liver samples. And our analysis looked at microcystin and anatoxin A, but we did find no anatoxin A, so I'm not going to discuss that further. So this is where we really started to encounter some of our analytical issues when measuring the toxins in biota. We first employed Envirologic microcystin LR ELISA. This was the ELISA kit used by the King County Environmental Lab, which is the state lab for measuring microcystins across the state, because it was good for water samples, but we had really low spike recoveries and inconsistent results. So at that time, we switched to Abraxas, which is now Eurofins, ADDA ELISA. And if you recall from a couple of slides ago, this is an ELISA kit that can target all microcystin variants. It has had more acceptable recoveries, and we used it for the remainder of the study. We also had the opportunity to send some samples out for LCMS MS to do a subset of our samples and look at particular microcystin variants that may be present. So on the left is a figure. These are results from the ADDA ELISA and yellow perch from Ketchum Lake. And the purpose of this table is to show how microcystin is highest in the liver, followed by the gut contents, skin, and muscle. And that happened in both of the fish that were analyzed from this lake. And then over here, on the x-axis, you have the ELISA results, and then on the y-axis, you have the LCMS results for 4 samples. And we had duplicates of each of these samples, and they were averaged. And as you can see, the ELISA was measuring much higher concentrations of microcystin in these fish species than the LCMS. We only detected microcystin LA of the 9 variants that we tested for. And the max microcystin in the samples was 12 micrograms per kilogram in a largemouth bass. That's what the LMB stands for. But over here for the rainbow trout, ELISA measured up to 140 micrograms per kilogram. It's unknown if there were variants that the ELISA was measuring here, or if there was some type of matrix effect that caused the ELISA to measure more of the toxin. We conducted a health assessment on our results using World Health Organization tolerable daily intake of 0.04 micrograms per kilogram per day. And we determined that children could safely consume up to 20 micrograms per kilogram and adults 40 micrograms per kilogram. The maximum microcystin in the fish muscle or fillet in our study measured by ELISA was 11 micrograms per kilogram, which led us to conclude that humans could safely consume 2 8-ounce fish fillet meals, which was the recommended health dietary guidelines, a week of fish from Washington state lakes during our study period. So our next question is, we stayed in the Puget Sound region, and we started hearing in the literature and stories about how there was microcystin contamination from freshwater bodies moving downstream and polluting the land-sea interface. This was first documented in the Monterey Bay area, where sea otters died from microcystin intoxication, and it was thought that the exposure route was by eating contaminated shellfish. So once again, here's Puget Sound. We had 4 study lakes for the project, and then up here, this Penn Cove was considered our control site because there are no toxic blooms feeding into that area. These areas all have lakes that drain directly into the Puget Sound, and people are recreationally and commercially harvesting shellfish in the area. And certain populations, including some Native American tribes, depend on collecting local shellfish for food and eat a lot of it. This was 2 years of research. We deployed cages of mussels at the mouths of the creeks discharging from lakes with toxic blooms, and we collected mussels weekly over 2 summers. We analyzed with ELISA and LCMS. We used 10 different microcystin variants. So this is another analytical issue or finding that I want to share. So based on the results from the Washington State fish, we wanted to investigate the use of ELISA kits a little bit more and make sure that we weren't getting the matrix effects or reporting false positives. So this y-axis is micrograms per kilogram. These are grocery store mussels that we assumed had no microcystin in them. So what we did is we compared 2 different extraction methods. A common method for extracting microcystin out of shellfish and finfish is using methanol. We did a long methanol extraction, which was 24 hours, and then we did a short methanol extraction, which was just a couple of hours. We ran those samples on 2 different ELISA kits. This is that ADDA ELISA that I mentioned targets all microcystin variants. And then during this study, I was talking to Abraxas and some of my concerns about the matrix effects, and they suggested that I use the direct monoclonal ELISA kit that they also produce. And this kit also is able to measure all microcystin variants by targeting that ADDA side chain. And what you can see is that my control mussel here, which should have had no microcystin, I was measuring up to 10 micrograms per kilogram. When I analyzed that same sample on the direct monoclonal, it was under 2 micrograms per kilogram. Using that same set of samples, different individual samples, but they came from the same batch, we did the short methanol extraction. And once again, the ADDA did measure significantly higher microcystins than direct monoclonal, but as you can see, we didn't have nearly the matrix effects that we had with that long methanol extraction. So based on this information, all of our analysis moving forward was using the direct monoclonal short methanol extraction. So this is an example of the results that we found. This is Bay Lake, and it's about a mile downstream to Mayo Cove here. This dark line is the microcystin that was measured in the lake, up to 180 micrograms per liter. Then this dotted line is the microcystin moving downstream in Mayo Creek. And then these circles is the microcystin in the seawater of Mayo Cove. And then these squares are the microcystin in mussel samples. We confirmed our ELISA results with LCMS and found 2 microcystin variants were present, microcystin LA and microcystin LR. So we detected microcystin at 2 of the 4 Puget Sound locations and in none of the control mussels. The maximum microcystin in the Puget Sound mussels over the course of the study was 15.5 micrograms per liter, or that should have been per kilogram, not liter. But so we had some shellfish consumption rate information from 3 tribes, Native American tribes in the area. And we took that consumption information and compared it to World Health Organization and EPA tolerable daily intake using both male and female. The 70 kilogram is the default value for males, and the 60 kilograms is the default value for females. These number in the far right column is the amount of microcystin per kilogram that one of these very high-end consumers. So these are, I should back up and say, these are the highest consumers of shellfish in these tribes, the people that are eating the most shellfish. And we calculated the safe amount that they'd be able to consume. And all of the italicized red numbers are numbers that if they consumed shellfish from our study, it would exceed what the safe recommendation was. And so you can see each of the tribes had some people that would be at potential adverse risk from consuming mussels during our study. This was just the compilation that we put together after that work was completed. we found, that in the Puget Sound region that microcystins can travel short distances from freshwater into marine environments, but we found that this is actually occurring in multiple lakes and reservoirs around the world. And up in Copco Reservoir in Oregon, California area, the toxins were found to travel up to 300 kilometers from the site of origin. And even for people that aren't working in marine and estuarine environments, I think what this shows is that the toxins can travel. And so if you have a river or creek, the toxins can travel out of that lake and move into different environments. Okay, and this is the last study that I'm going to discuss. This is a study that we just started in August. This is in collaboration with the Central Valley Water Board, San Francisco Estuary Institute, and Ben Genetics, and we're looking at microcystin accumulation in shellfish within the Sacramento-San Joaquin Delta. So cyanobacteria, harmful algal blooms, were first detected in the Delta in 1999. So just for a point of reference, here's San Francisco. We've got the Sacramento and then the San Joaquin River converging to form the Delta here. And then this outline is the area that is defined as the legal delta. A study in 2016 found that mussels out here in the San Francisco Bay were accumulating microcystins in all months of the year. Although the definitive origin hasn't been determined, one of the ideas is that it might be from microcystins that are forming here in the Delta and flushing out into the San Francisco Bay. There's also concern that Delta cyanobacteria harmful algal blooms are forming toxins that are contaminating Delta shellfish. There are several studies that have recognized this as a possibility, but it's really unknown how widespread potential toxin accumulation is throughout the Delta. So our project goal is we're looking at microcystin, and we're also going to be looking at freshwater saxitoxin. I don't have any saxitoxin results yet because we've only started sampling this August. But the purposes of this is that we want to determine if the toxins are stressors on food webs and native fish species, including managed species such as the endangered green sturgeon and white sturgeon that are benthic feeders. And we are defining it as a stressor if the toxin is present, because there are no guidelines on what a safe or unsafe amount of microcystin or saxitoxin is for a particular fish species. We're sampling for 2 years, 2 times a month during the main bloom season of June through October, and then once a month from November to May. Our target organisms are Asian clams, which are actually mussels. These are an invasive species that are basically blanketing the Delta. And then crayfish, which are also an invasive species, but they are used by a number of people in the Delta and organisms. They are sold to high-end restaurants in the San Francisco region. And I should note that the Asian clams, although they're really little, there are certain human populations in the region that do depend on eating them as an important part of their diet. And then we're also looking at water samples so that we can compare toxin concentrations in waters versus the shellfish. We're using the direct monoclonal ELISA following the short methanol extraction that I mentioned. So using very similar methodology from the Puget Sound mussel work. We're also running LCMS. We have a handful of variants that we're measuring, and we're also going to be able to do a subset of samples to look at the MMPB method, which will be interesting to measure total microcystins and all microcystin variants. This map shows the 10 sampling locations that we are going to be sampling during each of our sample collection dates. So these are preliminary results that I'm going to share over the next few slides. So far, we only have ELISA results, and we have results from August through December, which is 7 different sampling dates. Each of these sites, 1 through 10, correspond with one of the dots on the previous map. And then we have the amount of microcystin, micrograms per liter on the y-axis. And overall, I would say that the concentrations in the water have been relatively low, less than 5 micrograms per liter, and lower than that for most of the sites. 4, 5, and 6, you'll notice there's very little microcystin, and I'll show you shellfish next, and you'll notice a very similar pattern with the shellfish. Okay, so this is actually 2 of the exact same figures, but I removed the outlier from this figure for the figure below so they could see the scale better. So these are the Asian clams that we measured from the 10 sites. And we have lyophilized or run the samples through the freeze dryer. So this is reported as nanograms per gram dry weight. We had 1 clam over 1,000 nanograms per gram, but most of them, if you take out that outlier, were under 400 nanograms per gram. Based on EPA's tolerable daily intake, and then also including the dry weight factor, we assumed 18 nanograms per gram is a safe amount for human consumption. And so far you can see that many of our samples are above that safe threshold. We also looked at crayfish. We only have crayfish from 6 of the 10 sampling sites and a total of 14 samples. So I just combined them all for this preliminary view of data. We had 1 really high toxin concentration measured over 1,700 nanograms per gram. You take that outlier out and you can see the scale a little bit better. Still some pretty high concentrations of the toxin. So our intended outcomes for this project is we really want to develop an inventory of the accumulation in delta shellfish, identify which areas of the delta are most likely to have benthic contamination, and then define potential contamination of the benthic prey organisms. So in summary, I just wanted to go over some findings that I've come across over the last 10 years. Toxin contamination of aquatic biota is widespread. Microsystems are most commonly studied cyanotoxins in aquatic biota. Exposures based on my research and the research I reviewed seems to be most concerning for the highest end consumers, so the people that are eating a lot of shellfish and finfish that are accumulating the toxin. And we've made large strides in analytical work. We better understand matrix effects, extraction techniques, and the differences between analytical methods. And knowing which analytical method you're going to use is really important in determining actual toxin concentrations and what you're reporting. Yet there are a number of knowledge gaps. We need more field studies on cyanotoxins other than microcystins, freshwater saxitoxin and cylindrospermopsin. I wrote in here maybe anatoxin A. I'd be interested in hearing if people disagree with this, but based on what I know, anatoxin A does not persist. It doesn't seem to really bioaccumulate much in aquatic organisms, and I don't know that we need to focus a lot of efforts there, although I'm happy to talk about that, and if people have different opinions on that, I'd be interested to hear. We need more food web studies. How are the toxins moving through the food webs? What are long-term implications for this? Is this a stressor on species like what we're looking at in the Sacramento-San Joaquin Delta? That'll be important for just understanding how these toxins move through the food web. And then we also need to investigate the long-term chronic impacts of exposure to microcystins through diet. What about the people that are eating less shellfish? If they're eating it over a course of a lifetime, is there any risk associated with that? And then I would say, I think we've made big strides in the analytical techniques, but there's still work to do. Part of that is having studies report, their extraction efficiencies, including spike recovery tests, and just making that data more available so that we can compare what's going on across a variety of different aquatic environments. And I couldn't have done all of this work without all of these partners and different funding agencies. And so I just want to thank all of the people listed here and all of these different agencies and the people associated with them for helping with sample collection and just making this work move forward. Thank you.
Thank you, Ellen. That was great. We have several questions, and I'll start reading them and asking, are the fish kills mentioned on slide 4 due to toxin exposure or to the diel dissolved oxygen swings?
The fish kills that I'm aware of, there was up in the British Columbia, there was a net pen liver disease of salmon, and that was determined to be directly due to microcystin intoxication.
Okay, this question was asked before. I think your presentation came on, but what is your description definition of underserved communities?
That's a good question. I was thinking of economically disadvantaged communities, so people that are more disproportionately affected by environmental pollution. It can involve a lot of different communities.
And Carrie asked, will the higher tribal shellfish consumption rates be considered in either tribal or state water quality standards for toxins?
I don't know about that. That's Washington work. It was from a few years ago and it doesn't seem to have been incorporated, but I don't know what the status is of that anymore since I'm down in California now.
Yeah. Eric asks, are microsystems partially degraded during the process of bioaccumulation? If that is possible, does it change how you might think of potential toxic effects of consuming organisms that have bioaccumulated the toxins?
That's a great question. I don't know much about the degradation, like how the toxin changes as it passes from consumer to consumer. So if someone else knows, I'd be interested in hearing, or maybe that is something that warrants further exploration.
Right. And what is your microcystin recovery percentage in long and short methyl hydroxide extraction or methanol extraction?
You know, I would have to go back. I don't remember off the top of my head. I would be happy to share that paper with whoever asked that question, though. We do report that information in the paper.
All right. And then just one of the participants put in 2 URLs for OEHHA has fish shellfish tissue thresholds and FDA has them for cyanotoxin. So that's available for people who want to look at those links that are provided. Another question is, do you think there could be any difference in toxicity if the fish was cooked versus raw when the toxins were extracted from the fish tissue?
Yeah, there's evidence that the cooking could break some of those covalent bonds that I mentioned. And so perhaps more of that toxin would be freely available. And it's thought that that freely available toxin is the toxin that's more concerning of passing from one organism to another. And then also, microcystin is very persistent and it doesn't degrade easily. And so you can boil it and the toxin can stay intact. And so if you are making like a fish soup or something, you could potentially release that toxin into the soup liquid.
Is it safe to assume that grocery store mussels have no microcystin?
Yeah, we confirmed our results with LCMS, which, as I mentioned, they're not perfectly comparable, but I think in general, it's safe to assume that. That being said, there were some studies along the California coast that found the toxin in commercially harvested shellfish. So it's possible that it's there.
Yeah.
I don't think a lot of work has been addressed that, but it has been a known issue. In my particular study, I don't think there was toxin in those mussels that I tested.
Another question is BMAA still considered a potential neurotoxin?
I think so. That is not a toxin that I've spent much time investigating. There's an ongoing discussion in the literature about how important it is or how much it is related to cyanotoxins. I'm not an expert on BMAA, and it's not something that I've spent much time looking at.
Right. Max asks, would the outlier organisms be considered food safe based on morphology? And then he follows up, did they look healthy enough to pass food inspections, or would they look unhealthy and not be consumed in the first place?
They looked healthy. Yeah, there was no evidence of them looking different. And it's also, the bloom, this year's work so far was mostly outside of the bloom season. So it's going to be really interesting next year when we're able to capture more organisms during the height of the bloom season. That might not necessarily be an outlier, but for the preliminary data set, it is definitely an outlier organism, but no signs of anything wrong with the organism.
All right, we have a few more. What method was used to detect ANA in the Washington fish study? Did it potentially include covalently bonded?
It did not. It did not look at covalently bound anatoxin A. And I believe it was LCMS that they ran on that.
All right. Do you have an SOP or particular paper that details the ELISA method you use for finfish?
Yes. I have, so that box plot that I showed the different extraction methodologies, and then the comparison of ELISA kits, that has a full description of that. I'd be happy to share that paper with you.
Okay, so that person might want to contact you at your email afterwards.
Yeah.
Have you found matrix effects vary by species?
Yes, from my own personal work, I found a lot more matrix, and that same paper that I just mentioned looks at that. We compared a couple of different finfish fillets, and then also shellfish. And I was finding a lot more issues with the shellfish species than the mussel. The liver of fish also was having matrix impacts. And so maybe it's just more the lipids that are present in like the shellfish and the liver, are something about that is cross-reacting with the ELISA kits.
And then James asks, is there any research on toxin accumulation moving up trophic levels or outside the aquatic environment besides humans?
There have been some studies looking at it moving up the food web. I am less familiar with those, but there is evidence of trophic transfer.
All right, I think that concludes our questions. Thank you for your great presentation. We're going to have a break right now, and we want to be back at 1:55 Central Time to hear our next presentation. So thank you.
There. So, okay. So yeah, if you could just stay there. Okay. I'll wait.
All right, I have 2:55, so welcome back, and we'll continue the webinar. And our next speaker is Dr. Michelle Mostrom. And Dr. Mostrom is a veterinary toxicologist at the Veterinary Diagnostic Lab at North Dakota State University in Fargo. She received several doctorates, one in veterinary medicine from Iowa State University. And then after working in toxicology, she completed a residency at the University of Illinois and received her PhD from the University of Saskatchewan in toxicology. She did her clinical research on the environmental impacts of pollution in livestock in Alberta for numerous years, and she's done research in human allergics and asthmatics for several years as well. And she is a diplomate of the American Board of Veterinary Toxicology and the American Board of Toxicology. So welcome, Dr. Mostrom. The title of her talk today is Algal Blooms and Livestock, and you can go ahead and start sharing your screen and start anytime.
And how do I get here? How do I start that? Doesn't want to click. Okay, and is that working for you?
Not yet.
You don't see that.
No.
So we'll switch them back.
Okay,
There should be a little green button that says share screen.
yeah, I hit that. I hit that. So, okay. How do I get out of this? We're okay.
Then we'll move that to the screen.
Okay.
Then we'll go share.
Share.
Okay,
Then
now you're on,
I'm on, but
except it shows a Zoom.
okay.
So you want, yes, there. But now what you want to do is, right, display settings.
Now can you see this?
Now you're set.
With the help of IT.
and you can move this one down there.
Okay. I'm okay. Okay. Now I'm okay. You can see everything?
Yes.
Okay. This is a little bit different presentation because I'm not involved in research. I'm involved in the Veterinary Diagnostic Lab. And in the Fargo, North Dakota area. I've collaborated with Dr. Steve Ansley, who's at the Kansas State College of Veterinary Medicine, who's helped me out with some of the information, some of the cases and the slides, but all the errors will be mine. But I wanted to talk about in several areas that have been mentioned, and these are real problems for veterinary medicine. We do have issues with livestock, with food producing animals exposed to cyanotoxin blooms. And these blooms are not on the Great Lakes, or they're on the potholes in the prairie provinces and the upper Midwest. They're on the sloughs, the small ponds, the dams, the small lakes in the Midwest, where they occur and cause numerous livestock producers and veterinarians concerns about the quality of water and animal health. And these problems actually found us. We didn't go looking for them, but it began with numerous veterinarians and producers sending in water samples and wanting evaluation. And from there, we were charged with trying to give some interpretation. And I'm not specifically trained in cyanotoxins. I wish I was. I've had some experience with them, thanks to Dr. Beasley about 35 years ago on the University of Illinois with this. But it's been a continuing problem. And I'm not sure a lot of people are aware, but a large population of our livestock, our food producing animals, are dependent on potable surface waters. For cattle, once that calf hits about 3 months of age, she's not as dependent on the mama cow milk. They're looking for water sources, and they're looking for water sources until they're 6-7 months weaning, where they might go on more groundwater. But surface water is providing a large amount of the water to food producing animals, be it cattle, sheep, goats, horses, and swine too. And depending on the location, time of year, we can have blooms that are causing significant concerns and problems. And one of the first problems we run into, and which has been mentioned before, has been the producers just concerned with what is a bloom. And we get duckweed in because they think that duckweed is a bloom on their water. And that's one that's fairly easily solved looking under microscope. And we have producers out there that have drones, that have helicopters, they have fixed wing aircraft. They're checking their water sources maybe weekly, maybe every couple weeks. But as people pointed out, that's maybe not often enough for production of a bloom. And basically, most of the producers are just going on off-colored water as an indicator that they may have a problem. And they get veterinarians involved, extension agents help a great deal with water sampling. But that's our big problem is getting a good quality sample. About 70 to 80% of the water samples that come in to our lab, I think into Iowa State, K-State, are proactive. And they're asking a very loaded question. They want to know if that water source is safe to use for livestock, i.e., if I put the cattle out on this pasture with the water, is it going to support their production and not kill them? And the problem we run into is oftentimes you have no history. We don't know where that water source was taken. Sometimes they take water samples and throw it on the dashboard of their truck for hours, so the sample is compromised. Now we can get around those concerns, and we've tried with education. The extension agents and the veterinarians are much better at taking samples, but still, you want to know if that water sample is going to be representative of the water source. And sometimes we get in 5 to a dozen samples from one producer at a time where he's going to put cattle out on multiple pastures. So it becomes an issue for us and a liability because we assume the liability of safety when we respond in the report. And if we're wrong and kill cattle, then there could be a substantial economic issue for us. So I'm always conservative, but what our labs tried to do is provide a microscopic identification of cyanobacteria. Now that in itself can be fraught with some problems. Sometimes I can't identify them and send them out to a private lab for identification. I guess I don't feel cell numbers per mil is very helpful. We look at concentration in the water sample, but because not all cyanobacteria are toxigenic, we're not doing PCR testing here. I don't know if what I am seeing is potentially going to cause a problem, and then not all toxins are cell-bound. So are there toxins present there that I'm totally missing? I don't know if I have a bloom, what stage of the bloom may be. And in many cases, we see multiple cyanobacterial blooms going on. Microcystins are largely cell-bound, but that doesn't occur with all other cyanotoxins. The other issue we're charged with is that these livestock producers don't want to spend a lot of money for multiple tests. And they want, they may spend $50 to $100 if you want to do some ELISA tests. Hopefully you're doing ELISA tests for multiple toxins and not focusing on one and missing others. But they want quick turnarounds to move the cattle around on their pastures. And quick tests don't include all possible toxins out there. And they don't want to pay for the LCM-SMS test. So it becomes a level of the economics they're willing to pay, the risk they're willing to have for the testing, And then I really emphasize that it's just a snapshot in time. When they've talked, previous speakers have talked about the dramatic weather events causing blooms, which we certainly have here in the Midwest. We'll get 3, 5, 7-inch rains. We'll get bursts of high temperatures, or we'll get a snap of cold temperatures and then warm temperatures. So you really don't know where you're going to be with the water sampling. And you have to warn the producer of that. Keep in mind that a lot of producers that have large herds may only check them once a week or once every 10 days. So in some of our cases, we're way behind in sampling water samples that have been associated with some cattle deaths. We're not getting the true picture of what they're exposed to. And it's of real concern to the veterinarians and to the diagnostic lab to provide a good response to what the owners want. I put this in because I think in talking to the various toxicologists that do testing for cyanotoxins or cyanobacteria in water, we all feel that sampling is absolutely a critical issue to determine the cyanotoxins in the water sources and the potential risk for livestock. USGS has a very good publication out to use. I try to talk to producers about seasonal patterns, the environmental conditions of rains and winds and mixing, where they take their samples. Is it in a bay if it's a larger lake, where it is in the water column, particularly in larger lakes or reservoirs. And I've provided this picture to them for where to take samples. You know, if they can, if they've got a case where they have dead cattle and it's acutely, if they can go to the shoreline where the cattle walk down to the particular water source, they can see the tracks there. If they can take that sample, that's good. Cattle will go into the pools of water. They'll disturb the bottom sediment. They may want to take samples further out, but it's of concern where the cattle were actually drinking. And quite often we have no idea. And we may only get one sample with a case where we have dead cattle. And you try to talk to them regarding these issues, but it's a real concern. Again, veterinarians are much better, I think, about trying to get better samples for us. And the extension people are pretty good about it too. But we have some cases that have occurred here in North Dakota at the end of April, first part of May. The ice has barely gone out and we have blooms, and we have maybe a half dozen to a dozen dead cows out there. And we've actually had some research done on those with anatoxin A identified. And then our blooms will go right on through October, maybe into November. So the veterinarians are pretty concerned with the water quality and the potential for risk to livestock. I'm going to cover a few cases. The first case is in pigs, and you might not think of pigs as having exposure, but there's a number of swine facilities, confinement facilities in the Midwest, in Illinois, Iowa, Missouri, that are dependent on surface waters. And they have periodic cyanotoxin blooms throughout the year, and it can go into the fall and into the winter in the Midwest. And in this case, Drinking water in a swine facility, the surface water was naturally filled with from a watershed. The water was treated with hydrogen peroxide in a holding tank prior to use, which will not destroy toxin. But they were bringing in 5-week-old pigs and raising them up. And what they were seeing were these pigs had sudden death, very slow growth, distended abdomens, liver centrilobular necrosis, and areas of regeneration or necrosis with hemorrhage. And what's typical is you get a swollen liver, you get congestion, hemorrhage, you get some white areas, and that's where you're looking for liver damage associated with microcystin. The mortality in this group was increased by about 7.5% in 3 affected groups. Again, this was late fall and into the winter. Final mortality was about 11.4% in the pigs, but they had about 50% morbidity, which is huge, with pigs showing various clinical signs. The veterinarians went out there and walked the area around the surface pond and observed that there was a small area of standing water near the pond, and it had a little waterway to the main water supply, even in the winter. And they did determine that the water was positive for microcystins, LR and RR at 3.4 ppb and 2 ppb. They went to the standing water that they found and where they saw the algae growing, and it was positive for microcystins, LA, LR, RR, and YR. And that ranged from 3 up to 33.7 ppb for the various, that was for LA, but they found all concentrations for those congeners. They went back and tested liver tissue on some of the dead pigs and found the liver tissue was positive for microcystin YR at 1 to 3 ppb. That was done at Iowa State by LCMSMS. And that is from Dr. Inslee was involved in that one. But we have surface water as the main water source for several confinements throughout the Midwest, and this is a real issue that they have to deal with. As a toxicologist, that's my focus. I think about toxicology, but for veterinarians, when you have problems, toxicology is only about 8% of the problem, typically. So when you have losses in livestock, you're initially spending a lot of time ruling out bacteria, viruses, feed toxins, mycotoxins, and heavy metals, pesticides of other sorts, pollutants. For cattle that are dead around water sources, you might look at lightning strikes, and then you go to checking water quality and potential contaminants. So you can spend a lot of time and money as a veterinarian ruling in and out of causes. In the case of microcystins, they inhibit phosphatases 1 and 2A in the liver, and that causes the hemorrhage scene. You can see the gastroenteritis and kidney necrosis. Interestingly, microcystins can cross the blood-brain barrier and cause neurologic clinical signs also, which was in another case I'm going to describe. It wasn't in this swine case. WHO set a microcystin LR safe allowable limit at 1 ppb for humans. That guideline level is partially based on some experiments with growing swine. And we might see those low levels, but I think with livestock, typically we're seeing much higher levels because we usually have about, in livestock cases, some of the cattle cases, you may see half dozen dead cows, another 6 to 10 affected animals, and then the rest of the herd is off to the side. And you've got to consider with livestock, you're not dealing generally with a acute exposure. you're dealing with a subacute or chronic exposure to potential cyanobacteria in water. Part of the problem in the swine case was they were having fluctuating environmental temperatures during the fall and winter, causing the algae to die, release toxins, and then with warm weather, they had subsequent growth. So it was just the cycling growth of the algae. I'm going to move on to a sheep case. This was described in Australia, but it has an interesting comment here that I may relate to what another speaker said earlier. This was microcystis in Australia. 17 sheep died, and others showed signs of hepatogenous photosensitivity, which is liver photosensitivity. In the animals that did not die, they were monitoring recovery by serum biochemistry tests, liver enzyme activities, particularly gamma glutamyl transferase, and they also looked at bilirubin concentrations and found that they decreased fairly rapidly to normal levels in the 3 weeks immediately post-exposure to microcystis. However, they had an additional 34% of the sheep die during a 6-month observation period after the initial exposure, even though the serum biochemical tests gave no indication of the cause of death. And to the authors, it suggested that sublethal exposure to microcystins could cause prolonged morbidity and a delayed mortality and a more chronic effect. I don't know that I can comment from cases. Quite often in the cases that we deal with animals that have exposure to cyanobacteria, they're moved off the pasture, they're put on a different pasture, and eventually will go into the herd, and they're not kept separate from the rest of the herd typically for any follow-up. But when we have a significant concentration of cyanobacteria, my comment as a toxicologist is stop the exposure. If you can move the animals out without a lot of stress, that may be necessary for clean water, or fence out the animal source and provide an alternate water source, which rancher does not want to do. They do not want to fence out ponds or sloughs, and they don't want to haul water. So they want answers to how to clean up the cyanobacteria, which is not necessary. It's not out there for them at this point in time. For grooming animals, i.e. dogs, you want to bathe those animals immediately and remove algal debris. But that's specific for the grooming animals. The previous speaker talked about economics, and certainly we face that. Some producers have loss of pasture grazing when they have repeated blooms on pastures and cannot use the land as they would like to. They may have to be hauling in water, which causes economic costs too. And for veterinarians, when we have affected animals, we have to first consider what is the stress of handling these animals. If you have neurologic clinical signs in 1,500-pound cows, you don't really want to handle them. You may want to gently move them off the water source. You consider what medications you might use, and you have to consider whether they may be contraindicated with organ malfunction. Microcystin can lead to liver perfusion and liver failure. So you do not want to use a drug that requires liver metabolism and liver enzyme function. Microcystin LR can cause a change in the sodium transport in the kidney and kidney failure. So there's another concern for drug use. And you really have to be cautious and knowledgeable of what you're going to use in these food producing animals if you have compromised organ function. And of course, that's what the producer, the owner, wants for affected animals. If they're stumbling, ataxic, not eating well, have a diarrhea, they want them back to normal, and they want them treated, but often that isn't the quick answer. And another challenge for veterinarians. This was a case, Anabina derlicospermon, bloom in Southeast Oregon. And they had a very high death loss. They had 32 animals of yearling steers, 14-month-old steers that died in a 4-day period. And what the clinical signs reported in these steers were excitation, head tremors, staggering gait, tetany, and death. And most of these signs were neurological. Field necropsies by the veterinarians of several animals showed pale liver, and the histopathology was massive liver necrosis. So this is a case where you had microcystin causing primarily neurologic clinical signs. They identified 3,000 micrograms per liter microcystin LR in the reservoir water, and a very high concentration of 7,100 microgram per liter, or 7 ppm of microcystin LR in the rumen contents. And that work was done by LCM-SMS by UC Davis. So you certainly can have different clinical signs that you might not expect with microcystin. One question that has come up is how rumen microflora might provide a protective shield or a protective effect of hepatic effects of microcystin and nodularin. And one study was done where they incubated some of the cyanotoxins with rumen fluid from a fistulated cow at 3 different concentrations. They were 0.05, 0.5, and 5 micrograms per mil, which is fairly high, 3 hours in vitro. And these concentrations were based on what was suggested by acute dosage for cattle by the California EPA. They were analyzed by LCMS. Degradation by the rumen fluid of microcystin YR was 36%. Nodularin was fairly similar at 35%. Microcystin RR was degraded about 25%. And microcystin LR not very highly at 8.9%, and that was at the low concentration. There was a decreasing degradation at the middle concentration and no degradation of the microcystins at the high concentration.
Michelle, you have about 1 minute left.
Okay, the authors found that not much was degraded after that. We do have anatoxin A in dogs that very rapid clinical signs, neurologic signs. Treatment is of little of no benefit for those animals. You really have to get them, clean them off and get them in fast to veterinarians. Variety of toxins they've talked about, and that's why I'm so concerned for doing toxin testing in water, quick, effective. There's often the question is how do they control the bloom, and that has been mentioned with algicides. Using copper sulfate is not the answer, and copper accumulates in the sediments and is unsuitable for sheep because sheep are very sensitive to copper. So I guess conclusions, we really need a quick test that's fairly accurate for multiple cyanotoxins. And I think the long-term answer is proactive establishment of a riparian zone around our water sources to prevent the bloom from occurring. So that is an overview of what we face in the diagnostic lab. And it's a challenge for producers to try to manage these small water sources and for cattle and horses and sheep to be put out on them. Thank you.
Thank you very much, Michelle. There are several questions. We don't really have time for them. So we're asking if you could answer some of the questions that are in the Q&A section by writing in there during the next talk, if that's
Okay.
possible. And right now, let me just share my screen once again for our last speaker. And Michelle, again, thank you very much. And our last speaker is for this session, Tyler Butts. And Tyler graduated from St. Norbert College in 2018 and is currently a PhD candidate at the University of Wisconsin-Madison. His research interests focus on lake food webs and how they influence ecosystem responses to disturbance and ecosystem resilience. So I will stop sharing my screen, let Tyler take over. His title of his presentation is Food Web Structure Mediates Algal Response to Stormwater Event Pulses of Nutrient Loading.
Okay, is that up?
Yes, it is.
Easy. All right, cool. Great. I want to first thank everyone for letting me talk a bit about some of the research that I've been working on for my dissertation. With this talk, we've heard a lot of interesting talks about some of the negative impacts that harmful algal blooms can have on animals through cyanotoxins and as well as livestock and economic difficulties. But with this talk, we're going to take a little bit of a different tact and look at how animals themselves operating within a food web can potentially help mediate large algal blooms and cyanobacteria. And so the Midwest is increasingly experiencing infrequent high-intensity storms, which can produce these flashy pulse nutrient loading events that deliver large amounts of nutrients into water bodies, especially when large spring storms can coincide with fertilizer application. And this influx of bioavailable nutrients can then transition lakes from a clear water state over to a turbid water state that can often include a proliferation of cyanobacteria. However, ecosystems themselves are extremely complex, and the specific food web structure, the connections and makeup of species within the lake could potentially suppress or magnify the processing and cycling of these nutrients, depending on how that food web is established. And so just kind of go over this idea is in this, so there's a large storm event, there's this external loading where these pulses of nutrients are readily absorbed by algae that gets incorporated into algal biomass. Algal biomass is then potentially controlled or mediated by various food web interactions. A large interaction with algae is zooplankton grazers with a Daphnia pictured here, which in turn can be controlled by planktivores, which feed on zooplankton, setting up this classic trophic cascade structure. When I say that, is meaning that planktivores can suppress zooplankton grazers, which releases algae from predation and can contribute to larger algal blooms. However, these organisms are also taking in these nutrients, processing and recycling them throughout the ecosystem. And when we expand out just from this simple food web chain, there's many different components of the ecosystem where these nutrients are being allocated to, and organisms are actively recycling them in different parts, particularly when there's movement of between the pelagic zone and this middle open water and the benthic zone, which includes benthos, periphyton, and fish species that primarily prey on organisms in these different areas, as well as generalists like largemouth bass, which can move between these different areas and further connect and cycle nutrients throughout the ecosystem. And so this is to say that the food web itself is an integral component in how the ecosystem responds to disturbance, such as these pulses of nutrient loading, and can influence an ecosystem's resilience. And so when I say ecosystem resilience, a good conceptual way to think about this is a standard cup and ball diagram, that many people may have seen before, where the lake itself is represented by this ball, and some disturbance, some large nutrient load, moves the ball into and out of this saddle here. And depending on how the level of ecosystem resilience, how quickly those nutrients get cycled or get just moved all into algal biomass, the ecosystem can either stay in this clear water state, or in an instance where there's a low ecosystem resilience, it could be easily knocked into an algal dominated state. And food webs are one of the factors that can influence how resilient an ecosystem is through top-down control, as well as through, as I mentioned before, the cycling of the nutrients. And so we wanted to investigate this potential relationship by experimentally testing the degree to which the food web influences ecosystem resilience to disturbances such as these pulses of nutrients from the environment. And today I'll be talking about two different experiments that we worked on, looking at these ideas that try and link food web structure to ecosystem responses to storm events. The first will be looking more simply at just at top-down grazing pressure, and the second experiment will be looking more at how food web complexity can contribute to this idea. And so in order to test these ideas, linking food web structure and ecosystem response, we used large experimental ponds to simulate natural shallow lake conditions. These ponds were located at the Iowa State Horticultural Research Station and the Aquatic Research Facility. These ponds are extremely unique and useful. They are there's 6 of them in total. There's about 2 meters deep on average. They're naturally colonized by phytoplankton, zooplankton, and benthos, and they contain native populations of emergent and submerged macrophytes. And our experimental design to test this was a reference and nutrient addition design, where we established food web pairs, where one we left alone for the duration of the experiment, which over the course of the summer, and one we did a nutrient pulse perturbation, which simulated this storm pulse loading event, which ended up being about a 5% increase in phosphorus at about a 25-to-1 nitrogen to phosphorus ratio. And before and after this nutrient pulse, we did a suite of ecosystem monitoring. The main response variables that I'll be talking about today are, we looked at algal biomass at daily measurements. So we have a good record of the dynamics of algal biomass throughout the course of the experiments, as well as dissolved oxygen saturation, which is a good sense of how much oxygen is being produced and how comfortable organisms are within the water. And that was taken with loggers reporting every 30 minutes. We also measured zooplankton biomass and community composition 2 to 3 times weekly over the duration of the experiment. And so, or both experiments for both summers. And so first, I'm going to be talking about this first experiment that we did, looking at just top-down grazing pressure. And to do this, we used big mouth buffalo. Now, I'm going to be talking about buffalo a lot during this talk. And just to clarify, it is a fish, not the actual land buffalo. They're large planktivores, both in size and in ecosystem impact, as they can prey on a lot of zooplankton, mainly in these large grazers such as Daphnia. And you can definitely see how we're holding the fish. We're not a fisheries lab, but we make do. But in any case, as I said, they're large planktivores, and they kind of set up this simple food web structure where there's a planktivore preying on zooplankton, with zooplankton preying on phytoplankton. However, big mouth buffalo densities can be changed in shallow lakes through incentivized harvest. And these are contracts that allow for removal of big mouth buffalo through commercial harvesters, and which also help incentivize removal of some common carp populations that can grow to very large densities in many shallow lakes. And so with these incentivized harvest contracts, there we go, their densities can be reduced, potentially changing the strength of that trophic cascade and releasing zooplankton. But what I'm interested in is how does this affect how the ecosystem processes perturbations such as nutrient pulse loading, and are there implications for ecosystem resilience based on the density of big-mouth buffalo? And so what we want to do is put different densities of big-mouth buffalo in these experimental ponds. And to do this, we went to a shallow lake in Iowa that was also about 2 meters deep on average at its deepest. And we based the densities on actual ambient and post-harvest densities found in Iowa lakes. We collected the big-mouth buffalo, and we set up these 3 treatments of being no fish, a harvested density of big-mouth buffalo, as well as an ambient density, so a none to medium and high kind of density gradient. And we asked, does big-mouth buffalo density influence ecosystem resilience to perturbation? With the perturbation being a nutrient loading from a storm event. And what we would expect is that the ecosystem resilience would be along this gradient following this trophic cascade strength, where at ambient density, it would be lowest, and at no fish density, it would be highest. And in terms of what we'd be seeing from the ecosystem itself, we would expect a large increase in algae from an ambient density pond being pulsed versus a minimal increase in algae from a high or from a no fish pond, which would indicate high resilience. And so first, I would like to show you an average look at the community composition of zooplankton prior to this nutrient pulse. This is over about 5-6 weeks on average, the community composition. And we see kind of what we would expect is that there was greater Daphnia, so this large algal grazer in the ponds that had no fish. They were lower in the harvested and lowest at ambient density, which goes to show that the composition of the zooplankton was about what we would expect: high overall biomass and greater Daphnia biomass in particular. And so now let's look at how these ponds responded to that nutrient pulse. And so just to orient you from left to right, there's no fish ambient, so increasing big-mouth buffalo density. The green time series is the ponds that received the nutrient pulse, and the blue time series is a pond that did not receive a nutrient pulse. And the dashed line in the center there is when that nutrient pulse occurred. And we do see that there are differences in how algal biomass responded between these different treatments, with the reference ponds remaining fairly similar in all 3 ponds, or at least stable over the course of the time series. And what we see is that the algal bloom was proportional to big-mouth buffalo density. Peak biomass was highest in the ambient density pond, and it occurred the fastest within the ambient density pond, showing that there was this proportional response to big-mouth buffalo density. And one of the benefits of having these daily time series is that we can use statistical methods to answer questions about a regime shift, the movement from these different states that shallow lakes can move into. And one of those methods is called online dynamic linear modeling, where it uses an autoregressive model to take this time series and calculate eigenvalues. And when the green line moves above 1 from below, then we know that it's a clear evidence that a regime shift occurred. And when we look at these figures, we can see a clear and immediate regime shift at the ambient density. It moved above 1 from below, so that high density big-mouth buffalo. And there was evidence for flickering and then a later regime shift at the harvested density. So that lower density, but big-mouth buffalo are still there. And flickering just means, you know, that if you think again about that cup and ball diagram, the ball is moving back and forth between those 2 states, and then eventually settling into a new state. And so what this tells us is that with the regime shift data, it shows that there's also this proportional response to the big-mouth buffalo density. Moving now to the dissolved oxygen saturation, we saw that particularly in the ponds that received a nutrient pulse, there was a large drop-off and an anoxic environment created later in the season. But it was the timing at which this occurred was also proportional to big-mouth buffalo density, with the movement to anoxia occurring first at ambient density and last at the no fish density. Now, this wasn't just due to big-mouth buffalo. In 2019, there was a very large heat wave around the time that the dissolved oxygen started depleting, as well as it's known that increases in nutrients can lead to a increased decomposition of macrophytes. And so we believe that there's increased decomposition of these macrophytes that were depleting the oxygen concentrations within the pulsed ponds in particular, leading to this anoxia. But this just goes to show that that movement to anoxia occurred at this gradient of big mouth buffalo density. And so in summary of this experiment, we see, we can ask, does big mouth buffalo density influence ecosystem resilience to perturbation? And our evidence suggests that it did. There was a greater algal response, faster and more intense at ambient density. The drop in dissolved oxygen occurred fastest in the ambient density ponds, likely due to that macrophyte composition and just more algae decomposing as well that were in the system. And we saw a clear regime shift at ambient density, this flickering events, and then an eventual regime shift at harvested densities. And we did not see evidence for regime shift when there was no fish in the ponds. And so this kind of tells us that these harvested levels of big mouth buffalo are effective in, attaining greater ecosystem resilience. But this is also a pretty simple food web, and it's narrowing in. So we wanted to expand this out a little bit and look at some of those other food web components that I mentioned earlier in the talk, and kind of look at food web complexity. And the way we did this is by looking at benthic and pelagic components of the food web and how and the degree to which those are coupled. And what I talk about when I'm talking about benthic pelagic coupling, I'm talking about energy pathways that connect benthic and pelagic zones and lakes. So the pelagic pathway is what the big mouth buffalo experiment targeted, being algae, zooplankton, and a primary planktivore. In this case, it being a bluegill here, and the benthic pathway being periphyton, so algae that grows on surfaces, rocks, and macrophytes, preyed on by benthos, further preyed on by a primary benthivore such as yellow perch. And then these pathways can be connected by mobile consumers such as largemouth bass or fathead minnows that can move into these different areas and feed on different components of these energy pathways, which has been suggested to influence ecosystem resilience. And we hypothesized that a greater degree of benthic pelagic coupling would increase ecosystem resilience, as more coupling may absorb this pulse of nutrient loading more effectively and efficiently. And so we asked, can the degree of benthic pelagic coupling explain the difference in algal response to pulse nutrient loading? For brevity and for the sake of this talk, I'll be just talking about our low and high degrees of benthic pelagic coupling, with our low treatment comprising bluegill, yellow perch as our pelagic and benthic pathway components, and with a high degree of benthic pelagic coupling, including a largemouth bass, as well as fathead minnows at densities all akin to shallow lakes in Iowa. And so first, let's take a look at the algal response. Here, we can see the figures are the same. In this experiment, we actually did 2 nutrient pulses, which are designated by those 2 dashed lines that are vertical. And again, the green line is the pulsed pond and the blue line is the reference pond. And we see that at low coupling, algae responded faster to both nutrient pulses and attained greater peak biomass compared to the high coupling ponds. Whereas at high coupling, there was really minimal changes to algal biomass following the first pulse, and a slower response compared to the low coupling pond at following the second pulse. Going to show that there was these differences in algal response depending on the degree of benthic pelagic coupling that was there, providing some support for our hypothesis. However, when we applied that online dynamic linear modeling approach to this data in particular, there was no clear evidence for differences in the ecosystem resilience, whether that regime shift occurred in the algal biomass time series. And when we looked at dissolved oxygen saturation, there also wasn't a clear pattern or response there was just a general decrease over the course of the summer, but there also was no large anoxic event that occurred. And we did see an increase in dissolved oxygen saturation, likely commensurate with the algal response following the second nutrient pulse. And so what this tells us generally is that the food web is having an influence on how a lake ecosystem is responding to these external loads, such as these pulses of nutrient loading. In our first experiment, we saw that the density of the planktivore here really affected ecosystem resilience along a gradient of that trophic cascade strength, of the density of that top planktivore. However, when we expanded out, there was less clear of a picture, but there was still differences in the algal biomass response, depending on how coupled and how many connections between energy pathways there were in the lake ecosystem. So this is to say that we did find evidence that the structure of the lake food web, particularly in shallow lakes, which can be manipulated by fisheries managers, can play a key role in mediating the ecosystem response to storms and affecting how resilient those ecosystems are to external disturbances. I want to make sure that I acknowledge the many, many undergrads, lab technicians, and colleagues that helped me carry out this experiment the last 2 summers. With that, I will be happy to take any questions. So thank you for sharing your time.
Thank you, Tyler. There are a couple of questions. Have you considered the cyano-HABs might be coming in with the nutrient pulses from rivers, etc.? Conroy's algal loading hypothesis says this and has been tested in Lake Erie and Lake Superior and a couple of other places. Have you thought about manipulating situations where you could load cyano-HABs into your pond with your storm pulses versus where you do not see if the two different treatments differ?
Yeah, that's a very good question. I think that would be a very interesting experiment, and definitely, something that could potentially be done in the particular system that we had set up. In our particular experiment, these ponds basically had a watershed that we could control, and we were just focused and it's important to note that storms have a bunch of other effects to aquatic ecosystems, such as wind, the increased precipitation, hydrologic effects, and we're kind of focusing in on this pulse of nutrients into the system. But an influx of increased discharge from a river that's loading harmful algal blooms downstream into a lake would definitely have an impact on those systems. So that would be very interesting to look at the degrees to which that would affect our responses.
Great. Well, thank you, Tyler. I'd also like to mention that many of the questions that were asked of Michelle have been answered and are in the Q&A answered part. So if you want to view them, you can. Yeah, I think this last part was also with respect to Ellen's. So I'd like to thank Ellen, Michelle, and Tyler for their excellent presentations and very interesting presentations. I'd also like to make a few announcements. One, for everyone attending today, thank you for attending the symposium. Recordings of all these sessions, I know a lot of people have asked, they will be posted on the event website. It's in the chat box, northcentralwater.org. Please watch for follow-up emails on this and our upcoming HABs webinar series beginning in February. And then also in the chat box is a link to a short 5-minute post survey asking for your feedback on the overall symposium today and yesterday, and also some questions about future events. For those who have signed up for the final discussion today on animal health and HABs, the session will begin at 3 p.m. Central Time. And you should access the Zoom room link that you were sent, and then you'll be put into smaller groups. And so thank you again for joining us, and have a good rest of the day or a facilitated session.